Thank you for joining us here at the Renaissance Bio meeting. It's an update on the epigenetic clock of aging, and we've got three great speakers tonight, which Johnny Adams from the Los Angeles GRG or Gerontology Research Group. He'll be giving some additional background and intros of the speakers as we move along. But Renaissance is really just a group of local LA individuals, mostly researchers, scientists, or engineers and physicians that are interested in the biology of aging and in developing diagnostics and therapeutics that target fundamental aspects of aging and age-related disease. So we're very interested in epigenetics and the epigenetic clock of aging as a tool to help you track different interventions and also help interrogate different diseases and how epigenetics contributes to them. And I think epigenetics is often something that's thought of by most people as more amenable to modification or change with therapeutics or diet, rather than something like your genetics, which requires CRISPR or AAV gene therapy. So it's we're just kind of on the cusp of developing therapies that can influence your genetics and epigenetics. So we look at the epigenetic clock of aging as a pretty unique tool. So we're really happy to have some of the pioneers of it here today. So in additional point, there is a handout that I gave everybody here today. It's kind of an exercise. Renaissance Bio is intended to be a collaborative group, so people are contributing. We're not just here kind of watching a lecture or a seminar and then just going to go home and forget about it. We're trying to kind of work together to develop diagnostics and therapeutics and manage clinical trials and get data out there about these therapies. The handout, and so in kind of thinking about what would be a decent exercise to work on today, a collaborator of mine from Intervene Immune, Dr. Greg Fay, who's here, sent me an email from the NIH. They have a program called the All of Us study. So that's a 1 million-person study that they're monitoring people over time, kind of a personalized medicine initiative. And so the email kind of was asking, what should we track? Which is kind of surprising to me that they have this big bucket of money for an All of Us study and have done all this branding related to it, but they don't even really know what they're going to study yet, right? So one thing we think they should study is epigenetics and epigenetic aging. And so, one of the directors of it is kind of a tech guy, somebody formerly of Intel. There's a link related to it. He has a lot of kind of tech jargon about, how to develop different applications. So this is a use case scenario that he's trying to develop, for any given application. And but the handout just has the 7 or 8 different steps that he requires for each, and the people that are viewing online, there's a Google Slides presentation that we can all look at and edit in real time and contribute to this effort. The deadline for the submission to the NIH for the All of Us study is February 22nd. So we're going to be learning a bit tonight, and then editing and revising that use case, and then submitting to them within a few weeks. So please help out. Please try to contribute any ideas. And if you have any ideas after the meeting, feel free to shoot me an email, and we'll try to incorporate them. So I'm going to turn it over to Johnny to give the intros.
Hey, Bobby.
Thanks, Bobby. Welcome to the update on the epigenetic clock of human aging, and much thanks to Bobby and a lot of others for their fine work in making this happen. We are here at UCLA and being webcast around the world. So for those who are completely new to this, epigenetics involves modification of gene expression without changing the underlying DNA sequence. And the importance of this is often not well understood, even by senior scientists active in creating aging interventions in our community. And many recent studies demonstrate the connection between epigenetics, especially DNA methylation, and aging. And better understanding this across disciplines will allow us to better create solutions that will lead to better health and longer life. So all three of our speakers are outstanding experts in their fields. First up is Keith Booher. Now, Keith received his Ph.D. from the University of California, Irvine in 2011, where he conducted research in the field of cancer cell metabolism. He then began work at Zymo Research Corporation, developing methods and assays for the investigation of epigenetics. Along with colleagues, in 2016, he conducted a high-impact study evaluating methods for DNA methylation validation. And he continues to conduct research as service project manager at Zymo, with particular emphasis on epigenetics and the microbiome. And his presentation tonight will focus on Zymo's research and development towards new advancements and applications of the epigenetic aging clock. Keith?
Thank you very much, Johnny. I'd like to start by thanking the organizers for putting this special event together, and thank you all for being in attendance, and also those listening online. So my name is Keith Booher. I'm the Service Projects Manager at Zymo Research. So that involves all sorts of, epigenetics and microbiome analysis, as Johnny mentioned. But our newest service, which I'm going to spend most of my talk discussing, is this DNA epigenetic aging clock analysis. So we can take samples and assess their true biological age as opposed to a birth or chronological age. So for those of you who may not be familiar with Zymo Research, we've been around for about 25 years. Our headquarters is here in Southern California, in Irvine. We started investigating epigenetics in 2001, where we launched our first kits to help facilitate bisulfite conversion chemistry processes to do DNA methylation analysis. As the field of epigenetics grew, and so did our portfolio of epigenetics research projects, Zymo Research became known as the Epigenetics Company in 2008. And in 2012, we fully embraced the next generation sequencing revolution that was taking place in the field, and we launched a suite of services focused on epigenetics research analysis. Today, Zymo Researchers' services and products are the highest rated and most frequently cited for epigenetics research by the scientific community. And we've taken our years of knowledge focused on epigenetics and applied it to cancer research, new clinical diagnostics for the field of epigenetics, and also, of course, aging research is now a very big part of what we do at Zymo. So in addition to the roughly 185 experts, the employees of Zymo worldwide, we are also supported by a very well-respected board, scientific advisory board of epigenetics research scientists. So Dr. Peter Jones is a professor at the Van Andel Research Institute, director of research at Van Andel, and he studies a lot of the ways DNA methylation influences cancer and carcinogenesis. Also Dr. Steve Jacobson, so he is an expert in plant epigenetics and a distinguished Howard Hughes medical investigator here at UCLA. Dr. Alexander Meissner of Harvard University, who studies the connection between epigenetics, development, and stem cell biology. And also David Sinclair from Harvard University, a world-renowned expert in aging research. And Dr. Frederick Rue, who's a clinician at the Cleveland Clinic, advising us on clinical issues. So for those of you who may not be very familiar with epigenetics, I'd like to give a quick introduction, very short. I know Jim Watson will explain this in more detail. But first, some terminology. So genetics is the study of heredity, and our genome refers to all of an organism's hereditary information. Now, this information is carried in the sequence of DNA letters, which are represented by the chemical bases, which we call A, C, G, and T for short. Based on the arrangement of these sequences of bases, genes, gene promoters, and gene regulatory regions can be identified. So how is epigenetics involved? So maybe we can take an example. If we can pretend that the letters shown here represent the DNA sequences in our genomes, what we see is that when looked at in certain ways, we begin to see words formed. But when given specific grammar and context, these words carry a very clear meaning. However, these same words and letters, when given different context and different grammar, take on an entirely different meaning. So in a similar way, epigenetics can be thought of as the grammar that gives meaning to our genome. So all the cells of our body have the exact same genetic information, but in different contexts, they have different meanings. So whether we have the DNA is expressed as skin cells or gives rise to skeletal muscle, smooth muscle, neuronal cells, epigenetics helps to control this process. And so when translated from the Latin, epigenome means literally above the genome or above epigenetics, so controlling genetics. So the most thoroughly well-studied and abundant DNA mark, epigenetic mark, is the methylation of DNA. So this occurs on cytosine residues in DNA. And DNA methylation is often associated with gene silencing or turning genes off. Interestingly, so this is the chemical structure of these bases. A novel epigenetic mark recently discovered, DNA hydroxymethylation, or 5-HMC for short, is often associated with sites of active gene expression. So exactly how methylation silences genes or hydroxymethylation activates genes is not very well understood. So this is an active area of epigenetics research, but something that scientists at Zymo are trying to look at ourselves. So what we've done, this is original data from Zymo. What we see is that for a given DNA sequence, for fully methylated cytosines in that sequence, it seems to increase the thermostability of the DNA double helix. Conversely, the same sequence that's fully hydroxymethylated then has a relaxed or reduced thermostability. So what this means in plain English is that when you methylate DNA, it sort of compacts the DNA and makes it inaccessible to the molecular protein machinery, thus not allowing genes to be expressed. But unmodified cytosines or hydroxymethylated cytosines relax the DNA and allow the protein molecular machinery to get in and turn gene expression on. So it's important to remember that over the course of our lives, DNA doesn't change. So the DNA sequences we are born with, by and large, are the same sequences as we grow old until we die. However, epigenetics does change. So depending on the diet we eat, if we get enough vegetables, we stay away from fried foods, whether we smoke or don't smoke, if we get enough exercise and the right kind of exercise, we try to avoid stress-free lives. All these different environmental factors have direct impact and change and alter our epigenetics. So when you think about it philosophically for a moment, epigenetics really defines a mechanism in which our own personal experiences or life history is directly written into our DNA, which is the very fabric of the material that makes us who we are. So how did Zymo research get into aging? Well, this is as long as societies have existed, people have been sort of fascinated by this question of aging and the aging process. In Western tradition, aging is often, there's often a fascination, obsession with youth and how to prevent the aging process. This can be contrasted with Eastern tradition, in which aging is often associated with wisdom and therefore highly valued. But whatever the societal attitudes toward aging, worldwide, both East and West are soon facing a problem, societal challenges, as the average age of the population will increase dramatically in the decades to come. And so as shown by this Louis Cranach masterpiece here from Renaissance era Europe, he had this idea that, entitled The Fountain of Youth, the elderly would enter and then they would come out the other side sort of youthful and rejuvenated. So whether or not, a fountain of youth exists, if we could identify something like this would go a long way to benefit or solving some of these facing societal challenges. So you may think, well, of course, there's no such thing as this exists, and you would be right. However, revolution in the fields of molecular biology and biochemistry in recent decades has shown that healthy lifespan extension is definitely possible. The only caveat being that this is shown in model organisms, of course. So a couple examples, there's lots of research in this with yeast, C. elegans, all sorts of different organisms. One very interesting one from my alma mater, UC Irvine, is this Methuselah flies from Michael Rose's lab. Basically, what he did is a really simple experiment. So he just took the longest lived flies from each generation, males and females, and mated them to each other. And then he took the offspring from the next generation, took the longest lived, mated those to each other, and on for many dozens of generations. And what he found was that just by doing this simple experiment, he could get something like 4 to 5 time average lifespan increase for these flies. What's really interesting is not only are these flies long-lived, but they're also very healthy, very robust. So they're very active, they fly around a lot. If you introduce them into a population of wild-type flies, they quickly dominate the population, take all the food, and really just run wild. As the research has advanced, there's now more anti-aging research into higher, more complex mammalian systems. So mice studies are now a very active area of aging research. So a study released by Baker et al. A couple years ago in 2016 focused on senolytics and showed by modulating this, they can increase the lifespan, healthy lifespan of mice by almost 30%. So whether or not these studies can be translated into humans, the challenges faced by an aging population are definitely there. So it's known that aging is a major driver of chronic disease. This is important because this is very expensive for the healthcare system. So it's estimated that something like 75% of all U.S. annual healthcare costs can be attributed to the care of chronic diseases. This is also very deadly. So something like 2/3 of all deaths can be attributed to chronic diseases such as cancer, heart disease, diabetes, and neurological disorders, and so on. And I believe this chart from Cancer Research UK perfectly illustrates this point. What we see is that the age of diagnosis of cancer goes up a lot as people get older. So it's a little, yeah. I don't have one here. I'm not sure. So, and you can imagine a similar plot for one showing incidence of neurological. This is better, I think. Neurological disorders, heart disease, and so forth. So with these challenges, so the biotech industry has really sought solutions based on some of the fundamental biology that's been discovered in these model organisms in an effort to try to apply them to anti-aging interventions. So the idea is that they can reduce the rate of aging and help save healthcare costs and also offer health benefits to an elderly population. So a recent study estimated that even a modest 2.2 years life extension could translate to something like $7 trillion in healthcare savings over the next 5 decades. And so an article was published in Cell Press that featured some of the, it's about 3 dozen companies that are now, biotech companies that are now introducing some of these aging interventions. So these include things like just pharmacological targeting, so just administering drugs. So rapamycin and metformin, there's ongoing clinical trials and other similar compounds going on right now. So big data, so Google, its parent company, Alphabet, has invested hundreds of millions of dollars into looking at large data sets and trying to devise certain anti-aging interventions. Also, there's new direct-to-consumer, so if you go on Google and do some search for some of this stuff, you can see websites offering personal epigenetic aging coaches, for example, also vitamin or supplement regimens to try to slow down the aging process. Stem cells, of course, to regenerate older damaged organs. And one that I find very fascinating is this young blood movement. So it's also known as heterochromatic parabiosis. So the idea is that you can take the blood from a younger individual, say in their late teens, 20s, and transfuse that to someone who's older, and it's thought that there's all sorts of factors in the blood that can have these sort of more youthful effects on an elderly individual. And this was lampooned very famously about a year ago on HBO's hit comedy show Silicon Valley. So in this scene, we see this sort of Steve Jobs-like tech CEO. He's at this very important board meeting, and it's interrupted as he's getting his weekly blood transfusion from a younger individual. So whether or not any of these therapies will satisfy Kranek's vision of a fountain of youth is not clear yet, but the science around these therapies is based on real, rigorous, validated scientific data. And so what we need in addition to these therapies, is a good way to identify the best and most effective, because the problems facing society are occurring right now, these challenges with chronic disease, and they're only going to increase. And so if we had a good way to measure aging, we could identify the best interventions in an ethical and efficient manner. And so what we really need is a way to quantify aging. That way it can be measured. And so we're happy to say, so no surprise, we're all gathered here to talk about the epigenetic aging clock. This was first described by Steve Horvath several years ago. I won't go into the details, so I'll let Steve talk about this in the keynote. But just quickly, by analyzing the DNA methylation patterns from thousands of individuals, what he found is 353 key loci associated with aging whose, based on their DNA methylation levels, a biological age can be predicted. And so the idea is that based on these different methylation patterns, an individual could have either a healthy or sort of more youthful aging progression, or an unhealthy or sort of advanced aging biological age relative to the average or to the normal. And so this concept is illustrated further in this cartoon regression diagram here. So we see an individual from the age of 20, based on the type of lifestyle choices they make over the course of their lifetime, whether they choose to get enough exercise, eat right, get plenty of sleep, they could have either a very youthful entry into their senior citizen years, or if they make the wrong choices, they could have a more advanced age relative to their true birth age or chronological age. So when we saw this paper put out by Horvath, we found it extremely fascinating. So we've been at Zymo Research actively conducting all sorts of DNA methylation research and analysis. And of course, this question of aging is, it's immediately, something that just grabs you. And so we entered into talks. We contacted Dr. Horvath through some collaborations and work. We eventually decided to license this technology from UCLA and develop it further for research applications and commercial applications. So how do we study DNA methylation? So there's really one gold standard way to do this. It's based on this chemical process known as bisulfite conversion. So it's a multi-step process. I won't go into it, but at Zymo, we've been making kits and procedures to kind of facilitate this complex chemistry to make it very easy and accessible for the average researcher. And the end product is when you apply bisulfite to a DNA template, you can easily distinguish methylated from unmethylated cytosines just by looking at sequence data. So an example here is if you have a methylated cytosine in the original starting DNA template, you apply bisulfite, it's resistant to this conversion, and so when you look at the sequence data, it still looks like a C. However, unmethylated cytosine, it gets converted to T, and so when you sequence it, it's very easy to distinguish methylated from unmethylated cytosine based on the presence of either a C or T at that position. So Dr. Horvath's aging panel consisted of hundreds, 353 genes, and as he was publishing this research to the scientific community, Zymo Research, as well as others, had been trying to tackle a technical challenge, which was how to analyze many dozens of genes across many dozens of samples in a low-cost, high-throughput manner. So if you want to look at all of the methylome, you can sequence the entire genome, and you get all the information, but this is very costly. If you want to focus on a few genes, there are ways to do this too, classical Sanger sequencing, but this is low throughput and very labor intensive. So we developed a method based on microfluidics to try to increase throughput to look at locus-specific DNA methylation. So basically what this does is, we can design primers to post-bisulfite DNA for any sort of application. It doesn't have to be aging, could be cancer, developmental research, whatever. In parallel, we bisulfite treat samples. We then combine primers with samples in this microfluidics device, and we can process in a single run over 2,300 PCR reactions. We then harvest the samples, prep them for sequencing, generate the sequence data, and perform the analysis. We marketed this as our MethylCheck platform for locus-specific or targeted panel validation. And we were approached by a European group who was trying to assess all sorts of different methods to look at this sort of high-throughput locus-specific DNA methylation testing. And they said, would you like to participate? We said, of course. They sent us samples for which they knew the DNA methylation quantities, and said there were about 20 participating labs, of which Zymo was one, and said, choose your favorite method, measure the methylation levels, report them back to us. We'll kind of get an assessment of the best performing ones. So that study was published in 2016. It's a little hard to see, but it actually made the cover. It said DNA methylation assays benchmark. So we contributed our method, and what the authors found, I should say, is that DNA methylation testing, this panel validation testing, is now ready for clinical applications. Specifically, they found that absolute DNA methylation quantification, of which bisulfite sequencing is one, are the best methods, or the method of choice for these validation studies. And they concluded that with this new technology, DNA methylation should be widely useful now for early diagnostics, companion diagnostics, forensics testing, and other applications. So we were very pleased to see this in print, to see our ideas or thoughts validated by other scientists in peer-reviewed study. But with this, we were doing many dozens of genes in a single reaction, or at one run. But of course, Dr. Horvath's aging panel consisted of hundreds of genes. So we knew that even with this advance, we were not sophisticated enough to make the DNA methylation age testing accessible in a very high throughput, cost-effective manner. So we had to go back to the drawing board, come up with a new method, and we were eventually able to do so. And so we call this our SWARM technology, and it just stands for Simplified Whole Panel Amplification Reaction Method. So I won't go into very detail about how this works. We don't have enough time, but just want to say it's bisulfite sequencing based, it's high throughput, it's very flexible, so we can add or remove targets from the panel as the science evolves, and it's low cost, making it accessible to researchers. So I don't talk about the method, but I just want to show some example data. So shown in the panel of each figure on this slide, we measured the DNA methylation levels at these 353 highly informative gene loci for aging that Horvath described. And we cross-validated our bisulfite sequencing SWARM technology with array technology. So this older array technology was the type of data for which Horvath based his original studies. And what we see is that the two different technology platforms, they're very much in agreement. So it's near perfect correlation. Furthermore, we found that by switching to a more precise bisulfite sequence-based technology platform, we were able to reduce, so the original, if you're studying this with array data, Dr. Horvath published that the median error of the test was 3.6 years. But with a more precise technology, we can reduce that further to less than 2 years. So the platform is very robust. So here I just showed technical replicates. So there's near perfect correlation of these methylation values measured for technical replicates. So with that, plus some additional validation, we found that we had a robust platform to measure DNA methylation age. It was highly reproducible, amenable with very low input DNA methylation levels. So we've gone as low as 50 nanograms for certain applications. It's very accurate. It's flexible. As I said, we can add or remove targets from the panel as the science evolves. Very versatile, so it can be applied to different tissue types, and it's affordable because we're doing high throughput sequencing. So we brand this as a DNAge, and so we're offering this now commercially and with our research partners. And we were so excited about it that we decided to make it an emphasis and put it on the cover of our current product catalog, company product catalog. So we currently offer this test for DNA analysis of blood samples and also for urine samples. We're working on developing this for other tissue types as well. So currently testing for saliva is something we're close to finalizing and being able to offer. Other tissue types possible, skin, and so on. When we look deeper into the data, we start to see some interesting patterns emerge. So one thing we found that was really kind of fun is that we found that men will age faster than women. So I don't think this really comes as a surprise to anyone in this audience or listening online. For centuries and for societies across the world, we know that women have a longer lifespan than men. So it's very interesting to see this reflected in the DNA methylation information too, also. Specifically, when we dig deeper into that data, we find that the age acceleration seems to occur from about the age of 50, 55 years old. So surely there's some deeper meaning there. But what we begin to see emerging is this connection between health outcomes and environmental influences and aging. So what we've shown here in this graph, we looked at the DNA methylation age profiles of individuals known to be either positive for HIV or Down syndrome. So two conditions which alter or shorten an individual's lifespan on average. And what we see is that for all these individuals, they have a more advanced or accelerated aging profile compared to the population average shown here in blue, or a healthy average. Conversely, if we look at the same DNA methylation patterns in the children of centenarians, so people who live to at least the age of 100, and we look at the DNA methylation levels of their children, we see that they have a more youthful or slower aging profile compared to the population average. This is very interesting, especially in context of these Methuselah flies, these long-lived flies, which I mentioned earlier in the presentation. So with this test, we now have a way to connect health outcomes with the aging process. So for a long time, there's been all sorts of studies showing that different environmental or adverse health conditions alter DNA methylation patterns. So obese people have different methylation patterns compared to non-obese. Similarly, people that start up these focused exercise interventions have altered DNA methylation patterns in not only skeletal muscle, but also adipose tissue. People that smoke, there's DNA methylation biomarkers for avid smokers. Encouragingly, people who quit smoking, we see that their DNA methylation patterns can reverse. And this is also seen in, we did a study with the US Army looking at veterans coming after their tours of duty who have altered DNA methylation patterns that are associated with post-traumatic stress disorder. So with this DNH test and this epigenetic aging clock, we now have a way to connect all these different health outcomes with the aging process. So this is something we're very excited about, and the research is just growing.
Did you just test a bunch of people?
This is from publicly available data sets. So on the chart here, and I think also the healthy comes from our internal database. Okay, so I just want to close with one case study. So a particular application of the aging clock, not related to health, but actually for forensic science. So it's just a few slides. So we were actually approached by police detectives in Europe, Germany specifically, and they were studying a case. They found a suspect of interest in a criminal case whose fingerprints showed up in an international database of pirates. So they immediately notified the organized crime police of Germany, and they became very interested in this, and they wanted to, of course, prosecute or apply the law to this individual, this suspect. However, the age of this suspect was in doubt. So this person is younger, and like in Germany or Europe, there's different laws that can be applied to people who can be considered as minors or youth versus people who we treat as adults. So the detectives believe this person to be an adult, and they want to treat the law to the fullest extent. But the person said, no, I'm a minor, you have to kid gloves here. So they turned to us, because this person was a migrant or refugee, so there's millions entering Europe now, we see on the news all the time. So this is one person from this migratory flow. Reliable birth records for this individual did not exist. So they needed some way to assess the age of this suspect, and therefore they can apply the law. So they asked us for help. We said, yeah, of course we'd love to. This is fascinating. Send us the blood of the suspect, a blood sample from the suspect, and also send us, some control individuals. Don't tell us who's who. Let us measure using our DNH clock. We'll report the data to you, and then you can unblind us, and we can see what the results show. So they gave us 6 samples in total, 1 for the suspect and 5 sort of control subjects of similar age. So the data is shown here, but I think it's easier to visualize on this bar graphs. So the orange bar graph in each from each subject is their birth age or their chronological age, and then the blue bars represent our measured DNA methylation age. And then the error bars on each graph show the 95% confidence interval from our measurements. What we see for each of the 5 control subjects here is that the DNA methylation age is within the estimated confidence interval of the test. So this is sort of in agreement. However, when we looked at the suspect in this case, what we found is that our measured DNA methylation age showed him to be older than his claimed or supposed age. So it supported the ideas of the detectives. It supported their criminal case, and they're very excited about this. And they said, this is nice, but we really want to submit this into the criminal proceedings. I think we need to be more confident. So we suggested that they let us do repeated measurements of the subject's blood DNA samples, so we get a very precise measurement, and hopefully this can satisfy them. So we took 6 independent DNA measurements of this suspect's blood sample, and what we found is that we could now calculate a very tight confidence interval about approximately 27 years of age. So we estimate his biological age to be about 27. So this is, you don't need to run a statistical test. I mean, we did, and it is extremely unlikely that this person is as young as he claimed to be. So we're very confident about the DNA methylation age, or the biological age here, but we needed to take this one step further and associate that biological age to a true chronological age. So what we did was we analyzed subjects in our database. So we looked at males in the age range of this individual, so from 18 to 35, and we assessed their difference in their chronological age from their measured DNA methylation age, or DNH. So we get a delta age here. And what we find is that the vast majority of subjects in this age group were within about 2 years of their birth age. And for no individual that we measured was anybody showed a greater difference than 7 years. So based on this, we thought it's very unlikely that this person is as young as they claim to be. We created a formal report for this. I went to Germany a few months ago. I met with the detectives there, sort of presented this study. I signed the document. They administered it to the Office of Foreign Affairs. It's now in the public record. And I just got an email yesterday in which the forensic scientist leading this project said it's starting to make a lot of noise. Some news agency picked this up, and there's a lot of questions. There's a lot of forensic scientists very interested in this. Okay, so just to close, I just want to mention that we do offer this DNH test to research groups. So we have a clock, of course, for humans, but also now for mice as well. And for any consumers that are interested in testing their own either blood or urine, they can go to www.mydnh.com and order a urine or blood sample collection kit. And we'll send you the kit. You send us the sample back. We'll do the analysis and give you a report. We have specials in the back. We have a booth set up. There's a conference special, so you can take a coupon. If you're interested, you want to test for yourself, maybe just for fun, there's like a big discount associated with it. So anyone after the talk, please feel free. Okay, so once again, thank you for your attention. And if there's any questions now, yeah. So we'll have time for a few questions, I think.
I just have a quick question about the migrant. He presumably had a very stressful life when he appeared to be older than he was.
Could be, yeah, that could be. So that's why we wanted to look at the, as large of a sample cohort to see if is it common, or how likely is it that even if they live this very stressful life, could impact their.
Does it tend to affect younger people less? Have you
just
been finding
the
that?
psychological stress has really a very minute effect, and that would never affect
Especially when
it.
you're younger. but if you've
Yeah,
had a
just
long
repeating the
life.
question.
Yeah, I mean, you see an effect in a 70-year-old with 40 years of stress. That's where you see an effect. These short-term stresses wouldn't.
Sorry. So for those listening online, the question was if if this suspect in this case lived a very stressful life, would it impact their DNA methylation and sort of skew the results of the test? And Dr. Horvath kindly offered that psychological stress probably doesn't have a large impact on DNA methylation age.
One question was from online was just what is the cost of this? And somebody asked what's the cost compared to current DNA methylation, other methods. And somebody else also asked,
can they receive the coupon online also?
If you have their email information, I think we could work something out. We can get in contact with them somehow. So if they can send that to you, I can certainly get hold of it. The cost is $299. So that includes the kit, the collection kit, whether blood or urine. They just send it back to us. We do all the laboratory sample handling, generate the data, perform the analysis, and return the report. So the cost for the entire service alone is cheaper than if you were buying an array, for example, and that wouldn't come with any of the sample processing or analysis or DNA isolation. I don't not believe there's any other commercial entities offering DNA methylation age testing, so I believe we're the only one. There's a question, I think, over here first.
I just recently heard that they also test just protein-based test for methylation rather than gene. Does that make sense? Have you know about that?
Well, proteins.
Protein levels to detect assess methylation.
Yeah, but proteins certainly get methylated. A lot of proteins do. So the question was, does protein methylation?
Are there any tests?
Are there any tests for protein methylation associated with aging? So none that I'm aware of. There are protein tests for aging, but I think it's different than this sort of methylation modification that we're talking about. I think there was a question here first. Yeah, sorry.
It's a great talk. Thank you so much. My question is really, if I understood you, you have a very tight correlation, that straight line. It's very sexy. And you can actually predict someone's age based on their methylation, based on these 371 loci. Did I understand?
Yeah, that's correct. Yeah.
So my question would be, you're saying this is true throughout the body, all tissue types.
We can't say that. We can't say that necessarily. So this was developed for blood, and we're working on other tissue types. So the epigenetic, these DNA methylation marks, will be very different in different tissue types. I mean, we know this. And
I guess the question I'm really driving at is stem cell origin, right?
Mm-hmm.
So the question is, do old men, and that's where I thought you were going, do men over 100, when they have a baby, how methylated are their kids? 'Cause I come from the telomere measuring space, and I can tell you that there's no predictive value. The plus or minus 2 confidence intervals is everyone. So what you have is a real tight correlation, and correlation both ways. At what point does the epigenetics get reset? If an old woman or an old man has a baby, where do they start their methylation?
so this is really interesting. So this is a very active area of biology. So the DNA methylation patterns seem to get reset during zygotic formation, and as cells divide, you go from the 8 blastocyst stage to the morula stage and so on, gastrulation. DNA methylation patterns will change to push lineage specificity to muscle or neuronal lineages. So how these dynamics relate to the parental donor, we know they get reset, but somehow there's some memory that also goes with it. And I mean, how this occurs is a very active area of research. So please.
Okay, on the slide with the data, when you were talking about the last slide with data, the one with the okay, I meant the that one, yeah. Okay, so it looks like, would you say that there's another line implicit there, Down syndrome, HIV negative, and when the subject acquires HIV, they suddenly jump to another one of those lines, or is that?
Yeah, so the point I'm trying to, I don't know if the colors are a little off here, but or are difficult to see, but the idea is you can connect these environmental insults with a altered aging program. So We know that, I mean, you can imagine a similar one for cancer patients, people undergoing, whether it's the chemo or the cancer itself, also having this accelerated age. So it's you know, the stress of being infected with this, you know, very serious virus, or having a genetic abnormality on the body, which alters the aging process and either accelerates it, or if you have, you know, very good genes, or you know, very careful with your lifestyle, you can have a.
And those subjects, well, first of all, it looks like there's a much wider range for those, the HIV positive Down syndrome than there is for the other two groups. Is that?
Oh, could be, yeah, so probably the population average is gonna be the tightest, but the others, I mean, the sample size is smaller, so there's gonna be more variation, but the line should be, that should be the average value for that group shown on the plot. So we can see that they are different.
Okay, because some of them look like, some of them are well below the line and look like you would estimate their age in the normal population to be rather young.
Yeah, that's,
So
yeah, that's correct,
quite a
yeah.
bit younger than they actually
Yeah,
are.
yeah, it's very, very interesting, yeah. Please, in the back.
You showed that you've got the accuracy of the chronological clock now down to less than 2 years.
Mhm.
But I guess what I always struggle with a bit here is the more accurate you make it as a chronological clock.
Mhm.
I mean, imagine you had an absolutely perfect chronological clock.
Right.
There would be no biological information in that.
Right, so there's a,
So
yep.
are you aiming to get a perfect chronological clock for the sake of forensics, or do you want a clock that has biological information in it so you can tell things about biological age, predisposition to disease and mortality, etc.? What are you aiming for?
Yeah, that's a very astute question. So both, right? I mean, that would be until we,
But they're
yeah,
probably
in
two
the,
different clocks.
that would be two different clocks. It would be two different clocks. You're absolutely correct. So, you know, when we started working with this forensic case, we were using the, you know, the original biological clock, and you know, it seems to work very well. But if we could have a chronological clock, then like the question earlier, we wouldn't have to worry about this, you know, stress. Maybe they had a physical stress life as opposed to psychological, and that would impact for some reason. But if we had that true chronological clock, then for forensics, I mean, this would be the ideal clock. And then for other biological questions, we would have the, you know, the original Horvath clock, the aging clock. In the very back, I think.
Yeah, I'm wondering what you're measuring. Are you measuring genes that are unmethylated and genes that ought to be methylated that are demethylating at the same time? Are you looking at that kind of pattern, or just genes that are inappropriately methylated become solvents?
Yeah, so the question is whether we're looking at genes that should be methylated becoming unmethylated and how these patterns change. So I think the short answer is all that. So they should have a sort of normal aging or healthy methylation profile, and as we age and, you know, we become infected with HIV or diagnosed with cancer, I mean, these are going to, somehow they change as a result, and they're definitely influenced. So I think we'll do one more, or yeah.
Okay. So as a commercial product, you can offer a service and they can buy it and they can get a result. But what we haven't seen in the forensic case that you gave is the range of the data. So you showed that you can make the errors tighter by making one more samples, reading one more sample. But for any random sample, what's the mean error that would be associated with that?
So the median error for our clock is 1.9 years. Yeah. So for any one sample, it should be something in there.
Two standard deviations above.
No.
So you said plus or minus 2 years?
Yeah, that would be the median error. So if we take the average difference in chronological age from the methylation age, it's a 1.9 years.
That's not actually what I'm asking. So what I'm asking is, you take the same person,
Right.
and you measure that one person's biological age 100 times. What's the
Oh.
biggest difference that you'll see within that 100 samples in age?
Well, you know, looking at the data slide, it should be normally distributed, and most of the differences should fall within 2 years. About that, and you'll get the standard deviation, you know, differences set up in a, you know, normally distributed way.
So one of the questions we had online was about what cells are tested in the urine and blood test, and what is the overall agreement between those two tests?
Oh, yeah, so it's a different clock for, the answer is it's a different clock for blood and urine DNA. So we're just isolating whole DNA from either the urine sample or from blood, and then applying the bisulfite directly, and then looking at those panel of genes. So.
Okay. Are you aware of any data with the correlation of the methylation clock to Tregs, like recent thymic immigrant cells and?
We are looking at different leukocyte populations. That data, we're working on that. That data is being developed right now. So I don't have a solid answer for that yet, but active research. Yeah, at the
What
moment.
about correlation with telomere length or telomerase activity?
We looked at that some. There seems to be a big difference with the telomeres in our own analysis, and collaborators that we've looked at the same sample. There just seems to be a pretty big error with telomere. So the precision is.
Basically, there's no relationship, or very little of a relationship between telomere length and methylation age.
Yeah. Vince.
In cases of telomere lengths, we know that in some cases with aging, a certain segment of the population will actually get younger instead of getting older. That is, we'll get longer telomeres over a 5 or a 10-year period. Do we have any comparable data with regard to methylation as to whether this clock ever goes backwards?
We have
No,
some.
It does not.
Pardon?
It does not go back.
It does not go back.
It does
Thank
not.
you. Yes.
So, may I ask, in the urine samples, what cells are being tested?
Whatever cells would be shed into the bladder. Yeah, it's just isolating whole DNA from urine. So.
I find that chart depressing, because the best case is for the offspring of centenarians. One case, there's chronological age of 80 and they're 65. But all the others is just maybe the best 10-year difference for the offspring of centenarians, which I presume is the best case.
Yeah, it could be. Well, I mean, we'll see.
20, 30 years younger. That's sad.
I suppose, but if we can understand what makes these centenarians so young, if we can understand looking at these centenarians, they can give us clues to what's going on, what the biology is. And
It's kind of consistent with
Yeah, it's true.
So I think we've got one last question.
One last
and then
question.
we'll move on.
Thanks. So with respect to this question, the offspring of supercentenarians have about half the rate of cardiovascular disease that you see in the normal population. And so, two questions. One, what is the sample that was used to generate these data points? And two, might that be different from what you would see if you measured cardiac cells, for example?
yeah, that's right. I believe this is from whole blood. So the question was, if tissue specific. So if we look at supercentenarians have reduced rates of cardiovascular disease. So one is, what if you looked at the DNA methylation age of cardiomyocytes in these subjects, and also the first part of the question.
Yeah, so in other words.
Where did these samples come from? Yeah, so.
Just because the data are incongruous with what we know to be true. We know that supercentenarian offspring, at least those that inherit the right genes from their parents, will experience disease at dramatically reduced rates compared to the general population. And so epigenetically speaking, they must be much younger than the general population at the same age. And yet in this assay, they're not. So either that's because the cell that you selected for this assay is not representative of what drives that health difference, or we have a problem with the clock.
So these individuals are younger epigenetically.
I think of the former, yeah, exactly. But Steve, I know that you've said in the past that the different clots or different tissues all seem to give you the same result. So this may be a case in which they don't.
The age acceleration in one tissue can be very different from another. In other words, it could be that a person has young blood but old kidney or
Okay.
old lung.
Okay.
There's relatively little correlation.
Okay.
Okay, thank you very much for your attention.
Yeah, thanks, Keith. That was outstanding. Okay, James P. Watson is a physician and top expert on epigenetics and aging. He also posts detailed scientific information on a wide range of topics at the Anti-Aging Firewalls blog, along with our friend Vince Giuliano. And he'll discuss many important aspects of DNA methylation and epigenetics. Here's Jim.
Okay, I don't know if I can plug that one in. So you're going to just go ahead and Bobby, you'll just have to run the slides from there. Okay. So,
I'll let you struggle up there for a minute.
okay. Let's see, this one. Oh, this one. Do you mind if I use your back? Okay. Show up?
There we go.
Okay. Thank you very much for joining us here, for those that are here in person and those that are here with us online. I see a lot of familiar faces here in the crowd, and some of what I've presented here, some of you have heard before, but some of it is brand new, and I think you'll be very excited. I first should qualify, I am not an expert on epigenetics. That was a completely false introduction that Johnny gave me. Rather, I'm a plastic surgeon, and I can make you look a lot younger, but it will do absolutely nothing to your DNA methylation age. But we'll still shamelessly promote facelifts and many other things that have no usefulness in making you truly younger. So I guess the reason I got interested in this is people come in to see me in the office, and they typically say, Dr. Watson, which cream should I buy to make my face younger? And for many years I'd say, well, listen, I'm sorry to say this, but none of those creams work. And I of course, lost patients, and they were angry with me. And so, finally I started just asking the question, what really is aging about? And I got very interested in the subject, and that's when I met a friend of mine who's here with us today, Vince Giuliano, in the third row back here. And I really want to thank Vince for a lot of information I'm going to present to you. And we're going to shamelessly promote our website here too, so that if you want to read more about it. So, the other thing I wanted to mention, if I had, after listening to all your questions, if I had to change my title here, I think what I would put is a biological and clinical significance of locus-specific DNA methylation age. And that's very important. I'm going to bring it up over and over, because what you need to remember is your DNA has over 3 billion base pairs, and only about 21 to 22 million of those are actually CPG sites. Is that right, Larry? From what I understand, about 21.5, somewhere in there, plus or minus a few million. And only around a third of those are methylated. Isn't that right, Steve, normally? So, and the other thing I want to remind all of you, and this is really a very important point, when the oocyte is fertilized and becomes a fertilized egg, guess how many methylation sites are methylated? Anybody want to know? Except the people who really know. 0. And to me, this is the most amazing miracle in nature. And that is that when a baby is born, he's always 0 years old. And I want you to think about how profound that is. So let's take a 80-year-old dad, and the mom, let's say he robbed a cradle, he married a 25-year-old, okay. So you would think, if you're doing simple math, that the kid should be somewhere between 80 years old and 25 years old, right? If you're thinking the model that you asked in your question. But that's not how it works. It doesn't really matter how old the dad or the mom is, that the DNA methylation age and the age on their birth certificate is always 0 years old. And what Steve showed is that if you do in utero DNA methylation age, their age is less than 1 year old. So that tells you how accurate of a clock this is. So, we've looked at many other things as clocks. Telomeres are about less than half as accurate as DNA methylation for measuring age. Measures of cellular senescence are no better. H2AX, beta-galactosidase, P16-ANG4A expression. So right now, unless something better comes along, this is the best objective test we have. But it has to be locus specific.
What's CPG?
If you can wait until the lecture is done, there are 4 slides on that. So please, the questions are at the end, not in the middle, okay? So for more information on this subject, here's a couple references that are really easy to understand. I really like these. But if you want to go to the best reference by far, it's our reference. So here's the shameless plugging. This is a website that Vince started many years ago. And I sort of hitched on to his wagon and started helping him write this. And this particular one shows the blog we did on methylation. And it was posted on September of 2016. And here's the subjects that it goes over. And we just don't have time to go through all this. It talks about epigenetics in general, DNA methylation, demethylation, the 5 different telomere clocks. Dr. Horvath is only one of the 5 clocks that have been invented. And you can make a clock too if you want to learn how to do elastic net regression. It goes over comparing telomere length testing versus DNA methylation clocks in terms of accuracy. It goes over how Hutchinson-Gilford progeria syndrome is an example of accelerated epigenetic aging. It goes over Werner syndrome and the difference between Werner syndrome and normal epigenetic aging. It goes over DNA methylation in normal healthy aging without disease. It goes over the difference between epigenetic drift and epigenetic aging, which are different. It goes over the loss of inactivation, which is an epigenetic problem that occurs with aging. It goes over the loss of repetitive DNA silencing, which is an epigenetic problem with aging. And then it goes over some really weird stuff that Vince and I get very excited about. And if any of you want to talk about this afterwards, I'm happy to answer questions, but I never get questions on these. And that is the link between polycomb protein repressor complexes and DNA methylation age, the link between inflammation and DNA methylation age, the link between chronic stress, and I want to qualify, okay, we're talking about oxidative stress here, cellular stress. We're not talking about emotional or psychological stress. And then probably the most important thing is IL-6, a cytokine that we know correlates very well with the phenotype of aging, seems to drive CPG hypermethylation. And then cortisol, based on circadian cycles, not chronic stress, seems to also be a driver of DNA methylation. So we know that cortisol levels cycle every time the sun comes up and goes down. So one reason why DNA methylation clocks may be so accurate is it all depends on how many times the sun's come up and down during your lifetime. And you're not going to change the rate at which how many times the sun goes up and down every year. So that's why if a lot of this has to do with circadian cycles, you're not going to change how many times the sun comes up every year. And that's, in my opinion, one very important reason why this is a true measure of aging, and tells a forensic scientist how many times the sun has come up in that person's lifetime. So we don't have time to go through all that. So I'm going to basically stick to these 7 questions now. Instead of reading them all out, let's go right into it. So what is DNA methylation? Well, on your far left, there's a chromosome, and if you unravel it like a spool, you'll see there's tiny little spools inside the big chromosome. And those little spools are called nucleosomes. And each one of those nucleosomes has 2.7 wines of DNA around it. And along that DNA are cytosine residues. And the spool itself is made of 8 proteins that are all clumped together as a little ball. Now, you remember, sir, you asked about could you measure methylation in proteins. The place you might have heard about that was this structure right here. Nucleosomes have tails, and specifically several of these proteins, the most important ones is histone 3 and 4, have tails, and these tails get modified. The most common modifications are shown in this picture: phosphorylation, acetylation, and see the third one, methylation. So histones are proteins, and they get methylated, and that is part of epigenetics. So if you hear somebody saying, I'm measuring methylation of proteins, most likely they're referring to histone protein methylation. But DNA is not a protein, so we know if you're talking about DNA methylation, you cannot directly measure it. Now, is there a link between histones and DNA? Absolutely. It's just like the link between thread and the spool. And that's how DNA is compacted. So if you want to say in real simple terms, what is epigenetics? It's really 4 things, okay? It's gene regulation by histone protein modifications, not just methylation, but acetylation, phosphorylation, sumorylation, ubiquitination, and many other alterations to the tails of these histone spools. The second component of epigenetics is DNA methylation, and now we know it's not just methylation, it's also DNA demethylation, and that actively happens, not just with aging, but with memory formation in the brain. So DNA demethylation is not just a phenomenon of aging, it's a phenomenon of normal cellular function. The third component of epigenetics we're not going to really spend any time on tonight, and that's microRNA, and there's thousands of different microRNAs we know. And the fourth one is one that most people don't talk about much, but it's chromatin compaction. And that's probably the biggest reason why these DNA methylation sites are important, because they may not just be binding sites for methyl binding proteins, which we'll get into, but they may actually be very important for nucleosome binding. So in other words, it may be that the reason why the DNA winds around the spools is that certain DNA bases, specifically the cytosines, are methylated, and that, of course, facilitates chromatin compaction, which, of course, silences that DNA. So how does DNA methylation silence genes? Okay. Well, the simplest diagram up here is a cytosine DNA base, and you can see there's an arrow, which usually means an enzyme, right, a reaction, and then the methyl cytosine. If you notice, they always put the number 5 in front of it, and that's because it's always the 5' site on the cytosine that gets methylated. The enzyme that does that are called DNA methyltransferases. We're going to briefly talk about those. And the substrate, the methyl donor, is called S-adenosyl methionine, or the abbreviation for that is SAM. Some of you have probably seen SAM for sale in vitamin stores. It's often referred to as SAM or ESAM. It's been a big, popular supplement people take for depression. Some people say it works. There's not real strong evidence in clinical trials, but the bottom line is that's a methyl donor. So if you take a SAM or ESAM supplement, you're taking a methyl donor. Now, the methyl donor gets consumed, though, and the byproduct of DNA methylation is another small molecule called S-adenosyl homocysteine. And this is extremely important. If you don't remember anything else in this lecture, remember that S-adenosyl homocysteine is probably the boogeyman, not homocysteine, in a lot of the age-related phenotypes, and we'll get into that more later. Okay. So now, how do we put this together? On your left is a stylized diagram of a gene, and the little right-angle arrow there shows that that gene is being actively transcribed. Now, if you notice, there's a little symbol here, looks like a mushroom, for TF. That's an abbreviation for transcription factor. So as most of you know, transcription factor is a protein that usually migrates from the cytoplasm into the nucleus and then turns on a gene. But if that site is methylated, now the transcription factor cannot bind to it. Why can't it bind to it? Because when there's a methyl group, a protein comes along called a methyl binding protein that attaches at that methyl site. So really, the methyl binding protein blocks the transcription factor from binding to the DNA. So as a result, there's no DNA copying, no transcription that occurs. So now you know it all. You can leave now and go outside and smoke, and the rest of the lecture is not that much interesting. And when you come back in, you'll be older than when you went out. So again, TF is a transcription factor, and methyl binding protein is the protein that blocks the transcription factor from binding. Now, there's a lot more to methyl binding proteins than just blocking transcription factors. It may also be a protein that helps with nucleosome binding. It may also be a protein that helps with the three-dimensional architecture of the nucleus, how DNA attaches itself to the nucleocytoskeleton. But we don't have time to go into all that now. So, briefly, DNA methylation silences genes by creating a binding site for methyl binding proteins to attach to the methylated cytosines at promoters. This prevents transcription factors from turning on gene expression. So which DNA bases get methylated? Only the cytosines do. The adenines, the thymidines, the guanines don't get methylated. But it's even more specific than that. Specifically, if you notice in this stylized diagram, there's a number of cytosines here that didn't get methylated. Only specific cytosines do. And the ones that specifically get methylated, we'll talk about in just a second. So real briefly, there's three main enzymes that do this methylating. One is called DNMT1, one is called 3A and 3B. Of course, you're probably asking, where's 2? Well, there is a 2. It's just not as important. And then there's another one that has an L behind it. But the main important ones are DNMT1, 3A, and 3B. The methyl groups come from the folate cycle, SAM, which activates these genes, okay? And SAH, we talked about that earlier, is the byproduct of DNA methylation, and that inhibits the DNA methylation enzymes by feedback inhibition. So what's the clinical correlation of this? Well, if you don't have enough folate and B12, these deficiencies will prevent normal methylation from occurring. And this is why a lot of mental illnesses are associated with folate and B12 deficiencies, especially things like alcoholism and vegetarians who don't get enough B12 in their diet unless they're supplemented. But I hope that all of you don't think that you can solve this whole methylation problem just by taking some folate and B12, because there's no evidence that taking excess folate and B12 slows down or stops aging. In other words, if you have a deficiency, you need it, but taking mega doses of these vitamins does nothing to stop the normal epigenetic clock. You agree with that, Steve? Okay. So how does DNA methylation silence genes? Well, we talked about that earlier, and I just showed the transcription factors here, the methyl binding proteins which prevent the transcription factors from binding, okay? So now you know what transcription factors are. We need to briefly talk about promoters, because we didn't know. A promoter is a sequence in a gene that usually is on the 5' end of the gene. And that's just how DNA is measured, as what's the start and the end. And that's the site where the transcription factor has to bind, okay? So most of these sites that can be DNA methylated are located in the promoter area, okay? I should say the promoter area has the richest number of DNA methylation sites. And these are, of course, we talked about methyl binding proteins, and now I want to get to answering your question, and that is, what is a CPG? If you notice on here, I put, if you look at a DNA strand from the 5' to the 3' end, a CPG is where you have a cytosine and a guanine side by side. And now we're going to talk about that. So what is a CPG? A CPG is a cytosine right next to a guanine. And the guanine has to be on the 3' side of it, okay? The other name, CPG, is a cytosine phosphate guanine dinucleotide. So if you hear the word CPT dinucleotide, CPC, or just CPG, they're all talking about the same thing. So this is a CPG there, but this is not a CPG. On your right-hand side, this is just a CG base pairing. It's a C from one strand binding to the G on the other strand. So make sure you understand the difference. When we talk about CPG, it's two dinucleotide on the same strand, not on two opposite strands. Okay. Now, here's what's really amazing. Because of the fact that the C has to be side by side with the G, only about 1 to 1.5% of the entire human genome is made of this dinucleotide sequence. Now, if you work out the numbers, that works out to be somewhere between 20 and 25 million CPG residues. And not all of these are methylated. In fact, when you're born, remember, none of them, when the egg is fertilized by the sperm, all the methylation sites are demethylated. And so basically when in a fertilized egg, I believe it's about day 5 to 8, isn't that right, Steve? All the methylation sites are removed. And then during embryogenesis, sites are rapidly added in a programmed fashion. And that's how an old sperm and an old egg become a young embryo. Now, the interesting thing is that not all these CPGs are distributed randomly through the genome. Certain areas of the human genome have many more than 1.5% CPGs, such as promoters. Promoters have as much as 18% of their DNA is CPG sites. And those areas where CPGs are very rich are called a CPG island. And then there's other areas, such as upstream from the promoter, called the 5' UTR, that also can have CPG-rich areas. And then the area that's the most interesting to me is a type of repetitive DNA called allorepeats. Are all of you here familiar with that, or you want to spend a mention on that? Allorepeats, to me, is the most fascinating thing, because the only species that have allorepeats are primates. And so all primates share this repetitive sequence of DNA. Now, people who don't really understand biology like to call it junk DNA. You can call it that if you want, but there's a role for allorepeats, both during primate evolution, and there's probably a role for it today. But there's about 1 million of these allorepeats in the human genome. So because they are show increased CPG enrichment as a term, they probably play a very, very important role both in gene expression and a very, very important role probably in nucleosome binding. But we're not going to go into all that tonight. So methylated DNA is silenced DNA. We talked about that. And you can notice once it gets methylated, the nucleosomes now can get closer together. And so now you have compaction of the DNA. And that also impedes transcription. So transcription is not just impeded by methylation of a promoter, it's also impeded by DNA compaction. And it's very clear that DNA methylation plays a role in that. And that's why heterochromatin, the type of DNA that is not supposed to be copied, is always compacted like this, and DNA methylation is very important. So in summary, DNA methylation at CPG sites allow nucleosome binding and DNA compaction. DNA methylation not only silences genes, it silences junk DNA. An example of junk DNA is repetitive elements, pseudogenes and fossil viruses. How many of you are familiar with that? Did you know that you have thousands of fossil viruses in your genome? And we're not talking about, latent CMV or Epstein-Barr virus. We're talking about viruses that are no longer functional, they're extinct, but they're still in the human genome and are passed on from generation to generation. And I want to briefly mention the 3 types of repetitive elements that are important, because we're going to talk about these 3 in a minute, and that is lines, signs, and long terminal repeats. Now we need to talk a little bit about demethylation. So what is demethylation? Well, there's both passive and active DNA methylation. Here's where methyl groups can be removed from the DNA. Active demethylation is an enzymatic process. Passive demethylation occurs when DNA spontaneously undergoes deamination, and then later on gets modified by what's called base excision repair or nucleotide excision repair. Now, the methyl group isn't removed in one step, though. It takes a series of steps to remove this methyl group. So the enzymes that do this have a funny name. They're called 10-11 translocases. And there's 3 main TET enzymes in humans, TET1, TET2, and TET3. TET1 and TET2 are the ones that get the most attention because they're involved a lot in cancer, and they appear to be very important in the formation of memory in the brain. But TET3 is the one that caught my eye because TET3 appears to be regulated by the glucocorticoid receptor, which, as we know, is a protein that binds to cortisol. And cortisol, of course, is their circadian hormone that gets increased during the day and decreased at night if you go to bed. TET enzymes oxidize the methyl groups in 3 successive steps. And then another enzyme, TGD, removes the oxidized one. So the first step you can see is converting 5-methylcytosine to 5-hydroxymethylcytosine. So when they first started finding 5-hydroxymethylcytosine, people just thought this was an oxidative side effect of these TET enzymes. But now we know that 5-hydroxymethylcytosine itself is a epigenetic mark and plays probably a very, very important role in areas where there's active gene expression. In fact, the organ where there's the highest level of hydroxymethylcytosine is the brain. And it's very clear that there it plays a very, very important role in things like memory and cognition and things like that. TET enzymes are probably the way that non-random, locus-specific age-related demethylation occurs, i.e. the half of the clock that Dr. Horvath is going to talk about next. And TET enzymes clearly play a role in aging. They play a role in cancer and disease. But this diagram here is really not a very good diagram. This sort of suggests that this demethylation process is linear. In reality, it's really a circle. So if you look here, I like to call this the full circle of enzymatic CpG methylation and demethylation. So if you notice, you can take a methylcytosine. It can be oxidized to 5-hydroxymethylcytosine, and with one more oxidation step, 5-4-methylcytosine onto 5-carboxycytosine, and then it can be completely demethylated back to cytosine and make the thing. But if you notice in that diagram, there's a shortcut too, and that's the step in the middle there called base excision repair. So it's a lot more complicated than we originally thought. But the bottom line is you can see now how methylation and demethylation can be an ongoing process, and that's why it seemed to be so important in the brain. So what areas of CpGs are methylated in the DNA, and what areas of DNA are unmethylated? Well, here's a stylized diagram of a chromosome. And let's start for the far left. How many of you, do any of you know how many methyl groups there are, methylation sites there are on telomeres? Any idea? It's a trick question. Don't fall for it.
0.
That's exactly right. Why? There are no cytosines in the hexamer, the 6 oligo repeat in telomeres. So when we talk about DNA methylation, it has nothing to do with telomeres, and that's probably why, as Dr. Horvath pointed out, these are independent phenomena that we see in aging. On the other hand, subtelomeric regions are methylated because those are normal DNA. Centromeres have pericentromeric regions, and as you can see from here, they are heavily methylated. And then there are those repeats that we talked about earlier, line, sign, and long terminal repeats. If you notice there, they're almost all methylated, which means they're supposed to be silenced. Okay? Then a gene itself, if you can see, the gene body may have methyl groups. The CpG shore may have some methyl groups. But in most genes, the promoter itself has an area that's very rich in CpG sites that's called a CpG island. And not all genes do. Only about 70% of genes have a CpG island in their promoter. But those genes, normally that area is demethylated. So real briefly, the types of repetitive DNA, there's lines, that stands for long interspersed nuclear element. They have to be silenced by DNA methylation. Signs, those are short interspersed nuclear elements. Example of that is the allorepeat, the primate-specific repetitive element. And then long terminal repeats, I don't want to go into all the details in this, but it's another type of repetitive element. So what 5 changes occur in DNA methylation with aging? Now these are the general changes. There's a lot more very specific changes, okay? So here's an example of a young person's methylation diagram. And again, this is just a stylized diagram showing different components of the genome. And then an elderly person. So if you start on the far left, you can see this is an example of a sign. Is that right? Yes. So that's a type of repetitive element. And if you notice there's arrows going back, going both ways, that's because in some of these the DNA can be transcribed in more than one direction. Those are normally heavily methylated in a young person. But if you notice, most of those sites get demethylated with aging. So sign demethylation is a feature of epigenetic aging. The next diagram, starting from left to right there, you see is a stylized diagram for a CPG-rich gene. That's a gene that has lots of CPG sites in it. Those are normally demethylated when somebody's young, and then with aging, they get hypermethylated. A good example of that would be a tumor suppressor gene that when you're young should be expressed, but when you get old, it gets silenced. The third thing there is lines. Now, you would expect the lines to be just like signs, but they're not. They do undergo some DNA demethylation, but it doesn't seem to correlate with aging like signs do. And it's not exactly clear why. The next one is a CPG poor gene. This is a gene that doesn't have a CPG island. And if you notice there, it undergoes some changes, but not nearly as dramatically as the genes that have a CPG island. And then the last example there is a long terminal repeat, which is another type of repetitive DNA. So real briefly, the 5 changes that occur with aging is global hypomethylation. Across the board, there's less methylation when you're old versus young. The second is locus-specific CPG hypermethylation. I'm sorry, that's supposed to be hypermethylation, not hypomethylation. And then genes with CPG islands in their promoters undergo silencing. CPG poor genes increase their gene expression. And then repetitive elements, especially signs and long terminal repeats, lose their silencing. And so do the human endogenous retroviruses, those fossil viruses in the human genome. Now remember, these are generalities. There's exceptions to almost all of these. Okay, now how does folic acid, B12, and the SAM-SAH ratio affect DNA activity? Well, I want to briefly show you, this is a diagram that basically shows how folic acid and B12 are both needed for what most people call the folate cycle, which of course involves, the methyl tetrahydrofolate enzyme. And then of course that takes homocysteine, converts it into methionine. Methionine then is converted into SAM, and SAM then is the methyl donor for the DNMT enzymes that now can methylate DNA and then block gene silencing. Now this would be important, say, if you wanted to block repetitive DNA from being expressed, okay? So real briefly, SAM, we talked about this earlier, is a necessary methyl donor, okay? SAH, S-adenosyl homocysteine, is the byproduct of DNA methylation and blocks DNA methylation by feedback inhibition. And the SAM and SAH ratio is actually what determines the activity. It's not the absolute amount of SAM or the absolute amount of SAH. Now, if you look at these four compounds here, you see there's 4 enzymes that basically turn that into what we call a cycle. Every cycle has a rate-limiting step, an enzyme that limits how fast that cycle goes around. And for the methionine cycle, or this, what I like to call the methylation cycle, the rate-limiting enzyme is this enzyme right here. And I didn't spell it out, but that's basically S-adenosyl homocysteine hydrolase. So as you notice, that circle has 3 other enzymes. Those other enzymes are not the rate limiting factor, it's this one. And this is extremely important because we're going to give an example of a drug that is very, very common out there that can affect that enzyme, and that's been proven to be a longevity promoting drug. And so so please remember this. This is an enzyme that we're going to talk about in just a second that is affected by metformin. So real briefly, SAH hydrolase is the rate limiting step for this whole thing. It's not the amount of folic acid or B12, okay? Now, I want to give you some examples of specific things that can make your DNA methylation age go faster, things that make the clock tick faster, and things that slow it down. Factors that accelerate epigenetic aging, including folic acid and B12 deficiency, we already talked about that. Smoking and air pollution dramatically affect. Now, I don't remember who it was earlier who was trying to point out that if you had a clock that matched your driver's license birthday perfectly, it wouldn't be that useful because it really wouldn't change with interventions. This is a perfect example. There's now people who are looking at locus-specific sites for DNA methylation that change with smoking. So there may be in the future a DNA methylation smoking clock for smokers. And that, of course, would be one where using elastic net regression, you looked for CPG sites that specifically change with that lifestyle intervention. And you could do the same with exercise. You could find a locus-specific clock, in theory, for a good thing. Heavy metals also accelerate the epigenetic clock. Alcohol abuse, malnutrition do, oxidative stress, and here, of course, we're talking about stress on a cellular level, a molecular biology stress. And chronic inflammation also accelerate the clock. Obesity is probably the #1 health problem in this country right now. And there's some evidence that it's actually going to start to decrease the rate at which people are living longer, because now obesity has overtaken smoking as the biggest preventable health risk and health problem in this country. Chemotherapy also dramatically accelerates the epigenetic clock, and so do corticosteroids. There's also factors that slow down the epigenetic aging. Caloric restriction slows it down. So does methionine restriction. Exercise does. Sleep does. These 2 compounds are actually natural products found in many fruits and vegetables that are helpful. We'll get to the questions at the end, if you don't mind. The active ingredient in green tea, e.g. CG, the active ingredient in curry, curcumin, and the active ingredient in soy products also slow down the epigenetic clock. Fish oil and vitamin D have also been shown to slow down. And we went over some of the molecular mechanism behind this in our blog, but the one I'd like to talk about the most right now is metformin. Metformin has clearly caught the most spotlight in the last few years on its effect on aging. And there's actually very clear evidence that metformin is an epigenetic drug. The paper on this was published in April of last year, 2017. And I want to share with you a little bit about this, and this will be the conclusion of my talk. And that is how metformin can slow down the epigenetic aging and increase longevity. I want to preface this by saying for many years we thought that the mechanism by which metformin worked was its effect on glucose, and that was why it was good for diabetics. And then we said, oh, if you lower your glucose, it must make everybody live longer. But there was a paper that came out last year that showed that the longevity effects of metformin were completely independent from glycemic control. So in other words, they're not saying it doesn't work for diabetes, they're just saying its longevity mechanism is a totally different mechanism. I'm going to explain that to you right now. So you already saw this diagram on the right earlier. Okay, so SAHH is a rate-limiting enzyme in what I like to call the methylation cycle, but most people refer that as the methionine cycle. Okay, metformin activates this enzyme right here by a very, very circuitous route. If you asked me to guess how it did this, I would have never guessed in my wildest dreams of how it did that. And the pathway is what they call the AMPK LET7 H19 pathway. And I'm going to show you the date on this. This was not my research, but research that was published in April of last year. And this is the reference on it here. So real briefly, the net effect of metformin is you have less feedback inhibition of the DNMTs due to a lack of buildup of SAH, S-adenosyl homocysteine. So how does that exactly work? Well, here's the pathway here. So here's metformin. Metformin, we know, is an AMPK activator. AMPK, when activated, increases LET7. LET7 is a microRNA. LET7 then inhibits H19. H19 is a long non-coding RNA. And H19 then, by that going down, you have a decrease in inhibition of SAHH activity. So I know this sounds very, very weird, but I'm going to show you the data here. Okay, so when you treat cells in vitro with metformin, AMPK goes up. No surprise there. That's something we all knew. The next one's a little surprising. LET7 microRNA went up. Now, for those of you who are not familiar with LET7, it's a microRNA that seems to be associated with stem cells, pluripotency, and stemness. So it's a very, very important microRNA, especially in embryogenesis. And then when LET7 levels went up, the long non-coding RNA H19 goes down. So H19 is a long non-coding RNA that is associated with cancer. So this explains, to a certain extent, not only the effects of metformin on DNA methylation, but also explains why studies that they have done of diabetics who are on metformin compared with diabetics who were not on metformin, that the ones that were on metformin actually had as much as a 30 to 50% decrease in cancer mortality. So not only is this important for aging, it's very important for cancer as well. So here's more of the data for this same paper. And so basically you can see that they clearly showed the effects of metformin through this pathway that involves a kinase, a microRNA, a long non-coding RNA, and then an enzyme that gets rid of S-adenosyl homocysteine. I would have never guessed that this was the mechanism by which metformin works. So real briefly, metformin is an FDA-approved drug that could be used off-label as an anti-aging pill. And for those of you who don't know this, there's actually a big push right now by Nir Barzilai and a number of researchers around the country to do a large trial of 3,000 people with no diabetes to see if metformin increases health span and possibly lifespan. The study has already been approved by the ethics committee. The FDA has already approved it, and nobody wants to fund it. So until, and if you have any spare change, please give Nir Barzilai a call. I think he needs, is it 30 million or is it 100 million to do it? It's a lot of money. I checked and I don't have that much in my wallet yet. So that's the end of the my talk. Any questions now? Anything? Yeah.
Question, Jim. So you said that with aging, there's a general hypomethylation that takes place.
Yes.
But that the promoter sites become hypermethylated.
Yeah.
That's a little paradoxical.
Yes. And that's what was so confusing. When I first read Steve's paper, I scratched my head and I got a little bald spot here. I scratched it so much. Because half of his clock is actually DNA CPG sites that lose their methylation as a function of age. And half of them, it's actually not exactly half. Is it 60-40?
193 gain, 160 lose.
Okay, do the math. So 193 increase their methylation and 160 decrease. And that's why, as I mentioned at the very beginning, if I had to redo my title slide, I would put locus specific on there, because specific sites increase methylation, specific sites decrease. And, you know, and please don't get offended at me, Steve. Steve doesn't have a monopoly or a special dispensation from God for methylation sites that change. There's many of these out there. He just happened to use a really clever program to find a small group that you could use to measure that. But other people have made clocks with other CPG sites, but all of the clocks, to my knowledge, have CPG sites that increase their methylation and that decrease. The only one that doesn't is there's one clock that only has 3 CPGs in it. It's not very accurate, but nevertheless, you know, that's how simple you can make it. So that clock is probably a little bit more like an egg timer, whereas Steve's clock is a little bit more like an atomic clock. You know, so. There's a question in the very back.
Yeah, when you talk about the SAH level of enzymatic activity, SAHH, when the doctor measures homocysteine, is that a valid measurement for that activity? Is it, or is it just something that everyone thinks, or they?
You know, I'm going to be honest with you. I have studied homocysteine many times.
Repeat the question.
Okay. The question was the enzyme SAHH and its relation to homocysteine. Is that right? Okay. So real briefly, SAHH is an enzyme that converts the precursor to homocysteine into homocysteine. So it's actually an enzyme upstream from homocysteine. The real problem in medicine is that we've been measuring homocysteine for decades now, and we've given people folic acid, and we've gotten their homocysteine levels down, and it doesn't change their disease.
I heard that. So
Yeah.
I don't think it's really that's not that useful.
Well, what I'm saying is that we missed the point. See, we should have looked upstream. You know, much like Denham Harmon looked at the byproducts of oxidative stress and thought that it was free radicals the problem. But now we think that aging is far upstream and that the free radicals are the effects of aging, not the cause. So I think most people would say homocysteine is the effect of aging, is the effect of inflammation, not the cause of them. And we were trying to treat the effect, not the cause. And when you do that, you don't make a difference in the patient's disease. One more question.
Jim, are there any other promising therapies to turn back the epigenetic clock?
I knew you were going to try to trip me up here.
No, I gave you fair warning.
Yeah, you did. Yeah, okay. Johnny, you're asking a rhetorical question, and the answer to that is absolutely. There's people in this room who are working on solutions to epigenetics. I can't discuss those right now, but I can tell you that I'm extremely excited about the epigenetic interventions. And I would like to take issue, in all due respects, Dr. Horvath, with the statement that the epigenetic clock cannot be reversed. I can tell you that on anecdotal cases, it can be reversed, but we don't have enough data right now to publish. But I do believe, and I will make a bold prediction, that in the next 5 years, somebody's going to show that an intervention will not just slow down the epigenetic clock, but will reverse it. I don't think it's going to be a magic bullet, because I don't believe that aging is a magic bullet deficiency. Aging is a multifactorial puzzle with maybe 500 pieces, maybe 1,000 pieces, maybe 100,000 people. But there's no question that epigenetic aging is a significant part of aging, and it's the best biomarker we have right now. So why don't we run with it, see if we can do epigenetic targeted therapy. And I am very optimistic that there are several things in the pipeline that will reverse the epigenetic clock.
Another question real quick. Is there any evidence for intergenerational changes from diet that you're aware of, or smoking?
Yes. I can tell you that you're saying, let me repeat the question. The question is there any evidence for intergenerational changes, that is changes from one generation to the next, right? As opposed to intra-generation change,
Like
which
if
would
you
be just in me.
while your kids
Yes.
have passed
Yeah.
away.
The answer to that is, I think, very important. That is, for a long time, we thought that the only way that information could be passed down from the parents to the children was by DNA-based pairs, in other words, the DNA sequence. But we now know that epigenetic marking can be passed from generation to generation. And there are 3 classic examples of that in history. The first was during World War II, when there was an area of Holland where a large number of people underwent a 9-month famine. And if you want to look it up, it's called the Dutch hunger famine. And that altered the epigenetic profile of the babies that were in the mother's womb who suffered that famine. And they have now documented that the effects of that have been passed on to the children and the children's children. The second example in history was in mainland China in the 50s during the Cultural Revolution. And during that time, due to the nationalization of the farms, and they made the farm collective, there was also a large famine that was not just due to the governmental the governmental policies, but also because of a large flood. And there were many more people who suffered malnutrition than the Dutch people in World War II. And in the Chinese example, not only did they show passing on of epigenetic predisposition to disease from one generation to the next, they also showed that the epigenetic consequences of that were an increase in mental illness. And that was very, very interesting to me. Both schizophrenia and bipolar illness increased dramatically in the offspring of the mothers who suffered malnutrition. The third example is one that's a little bit completely off base, so I won't go into that. Yeah.
If babies embryos or babies, let's say start out with 0 CpG methylation,
Yeah.
then that there must be a complex mechanism, let's say possibly involving protein methylation that passes on that epigenetic modification to the children at a later age.
I think you're right, but the bottom line is, in my opinion, that phenomena that the entire genome gets demethylated and then it gets remethylated to be 0 ages old, well, actually 0 minus 9, T minus 9 months, is the most amazing thing in nature. And I don't think even the experts in this field can completely explain what happens between day 3 and day 9 of the embryo, which actually at that point is still a blastocyst. So it's during the blastocyst phase where the entire genome gets demethylated and then gets remethylated. We know that DNMT1 is involved with that, but I can't tell you much more than that in terms of how it does. Is it involving protein? Of course, DNA methyltransferases are proteins, but exactly how that clock happens, we don't know. Now, we do know the driver of that demethylation, remethylation, and that's the Yamanaka factors, the pluripotency factors that are responsible for stemness. OX4, SOX, CMYC, KLF, NANOG, those factors.
One last question, we'll take a 5-minute break, but someone asked about cross-referencing DNA methylation with histone modification-based markers, and I thought you mentioned something at the very beginning.
Yes,
about
that's a very good question. The question is there any cross-referencing between DNA methylation and histone modification? And the word that I think the scientists like to use is crosstalk. There's a lot of crosstalk between epigenetic DNA methylation and histone epigenetics. And it appears in some cases histone modifications affect DNA methylation, other cases DNA methylation affect histone modifications. But exactly which is the chicken and the egg is not exactly clear. And what's even more interesting is there's a very evolutionarily old method of epigenetics that isn't really considered to be a separate area called polychrome protein silencing. And that's actually an area that I'm really interested in because polychrome protein silencing is much older than histone-based silencing and probably older than DNA methylation-based silencing. And polychrome protein silencing of genes seems to be very, very linked to the epicenter of cellular senescence. And so I would say that there's more than just histones and DNA methylation. There's also this evolutionarily old system of silencing genes called polychrome protein, polychrome group based silencing. So there's multiple crosstalks. I'm not sure if that answered the question.
Thanks a lot. All right.
Okay, thank you.
Hey, Jim, that was so clear. Could you tackle Bitcoin during the break?
Okay, everybody, we're going to start, and we're online, so we want everyone to be able to hear our next speaker. Steve Horvath is a UCLA professor and a pioneer, of course known for developing the Horvath aging clock, and also for developing weighted correlation network analysis. Now, he's the recipient of research awards, including an Allen Distinguished Investigator Award, and Dr. Horvath has studied genomic biomarkers of aging, the aging process, and many age-related diseases and conditions. And tonight, he will describe recent versions of epigenetic clocks and their applications. So here's Steve.
Thank you.
Thanks.
Thank you so much. Thank you, Johnny Adams, for organizing this wonderful event, and also Bobby's work. He did it all, and also Dr. Jar from Zymo. Thank you for staying so late. I will talk about updates on the epigenetic clock. So the original epigenetic clock was published in 2013, and there have been some developments. You've seen this slide before. We keep talking about the aging clock, but what the original multi-tissue epigenetic age estimator can do is it can measure age from prenatal samples all the way to centenarian samples. And it can do that in all sources of DNA, all cells and all tissues. And so what this clock does is it really links in certain ways development to maintenance during adult life to aging, this continuum, which is really gives us a profound insight into the nature, at least of epigenetic aging. So aging, is it really aging, or is it a continuation of development and maintenance, Or conversely, does aging happen already very early on in life? One thing I want to show you are some data from Akina Hoshino and Thomas Rhee from the University of Washington, where we applied the epigenetic age measure to fetal retina samples. And when you carefully look at the x-axis, you see negative numbers. So this is units of years, so minus 0.5 years, So we're talking here about gestational weeks, And in the retina, we observe correlations of 0.89. It's just striking that the epigenetic clock even applies to these very early phases in life. One thing that was mentioned is that this methylation clock does predict lifespan, And I should mention there's a huge error bar, So yes, it can say, well, you will live another, let's say, 20 years, but this association is statistically significant, but there would be a large 95% confidence interval, Why? There are other predictors, much more mundane, that are actually much stronger predictors of lifespan. One would be, do you smoke? Smoking status, or your blood pressure, So the epigenetic clock adds to these traditional risk factors, but it tweaks these age estimates a bit, and that is no longer, when I first published the paper in 2013, I just didn't dream that this would be the case, because it's almost too good to be true. But nowadays there are really half a dozen large-scale studies that have shown that yes, when we analyzed blood samples collected in the 1990s, and we had follow-up information on everybody, how long did they end up living? We knew, or who is still alive. So then, based on the blood methylation data from the 1990s, we could predict how long somebody lived. However, and I should say that it was already mentioned, there are several epigenetic clocks, and pretty much most of them have that property that they predict lifespan. But we and have worked on newer versions of the epigenetic clock that are more predictive of lifespan and health span. And this project was done by first author Morgan Levine. So we have developed a new clock which we call DNA methylation phenotypic age, as opposed to chronologic age. That's the idea, you know. It's in certain ways a better measure of biologic age for this purpose, and it also uses again hundreds of CpGs. In this case, 513 CpGs, so more. But it is quite remarkable what it can do. So this new biomarker can again predict lifespan due to all-cause mortality, but also it can predict what is known as cause-specific death, death due to cardiovascular disease, coronary heart disease, death due to cancer or diabetes or lung dysfunction. So it really covers the whole spectrum of age-related mortality and morbidity. But also, we find that this DNA methylation pheno-age is a predictor of coronary heart disease, that death due to coronary heart disease, which is quite remarkable because the previous version of our the epigenetic clock was not very predictive of coronary heart disease. It also correlates with the coexisting morbidities. So if you go to somebody and count their ailments, that is the morbidity index, and so the phenotypic age in blood correlates with this morbidity index, and also it relates to problems with physical functioning and also interesting cognitive functioning. So by now we're used to the successes of the epigenetic biomarkers, but 3 years ago, 5 years ago, if you had said that a blood measure on the DNA could relate to cognitive functioning, I mean, nobody would have believed it. You would never have gotten funding for that type of study because a reviewer would say blood is not relevant for the nervous system. I want to come to accelerated aging diseases, and Jim Watson already talked about it a little bit. So for a while, it wasn't clear whether Down syndrome should be considered an accelerated aging condition. But there's no doubt the epigenetic clock says that yes, blood samples, brain samples from individuals with Down syndrome are epigenetically older than age-matched controls. So the epigenetic answer is clear. And by the way, Down syndrome is one of the conditions with the strongest pro-aging effect. You know. And also in more recent work, we've worked with Anna Meyerhofer and George Martin and Thomas Haff and Junko Oshino to analyze Werner syndrome. And again, in blood, we found age acceleration in Werner syndrome individuals. Werner syndrome is known as adult progeria, so it has an onset in adult people. But there's another progeria, an accelerated aging disease called Hutchinson-Gilford progeria. And this is a condition that's widely known because it actually affects, unfortunately, children who manifest aging phenotype very early on. And many people have looked at, I've worked very hard on that problem to see whether I can find accelerated aging effects in Hutchinson-Gilford progeria. And I analyzed data, fibroblast data from the Progeria Research Foundation. And when I just applied my original clock, it doesn't find an age acceleration effect in these fibroblasts, you know. But there's a caveat, because if you ask me in what cell type does my clock not work well, I would probably start with fibroblasts. I mean, it kind of works, but you've seen these beautiful correlations of 0.95, you know, and we observe correlations of 0.9 and more in pretty much all cell types and tissues, but not in fibroblasts, okay? There the correlation is maybe 0.6, 0.7, you know. So then I've been thinking, well, maybe my original clock is deficient, so build a new clock. And so I've just, over the holidays, developed a new epigenetic clock, which I don't have a good name for, but I call it the epigenetic clock for skin and blood cells. And it really applies, as the name says, to cells in the skin, certainly fibroblasts, keratinocytes. It also applies to endothelial cells, but it also is phenomenally accurate in blood cells, but many other cells and tissues. However, its raison d'etre is really to facilitate these in vitro studies, because like many of you, I'm trying to find anti-aging interventions. And so what we have in mind is having a dish with some cell cultures, it could be fibroblasts, keratinocytes, endothelial cells, and then we try different anti-aging compounds. And my close collaborator, Ken Raj, has already tried several compounds, and some of them have striking effects on these fibroblasts. So coming back to Hutchinson's Guilford progeria, so I now applied also the new epigenetic clock to these fibroblast samples from the Progeria Research Foundation. And no matter how I analyze the data, I don't see an effect. So the right panel shows how this methylation age relates to chronologic age, and the colors indicate disease status. So red is classical Hutchinson's Guilford progeria, and green non-classical, and we really don't see a difference. So this is in certain ways perplexing, but basically HGP, according to the epigenetic clock, and in fibroblasts, there's no age acceleration. Could be in other tissues, who knows. I now want to come to another topic. There are, of course, several methylation-based biomarkers of aging. I talked about the multi-tissue methylation age estimator. Hannum and from the lab of Eidecker and Kang Zhang developed another age estimator for blood, but it also works in other tissues. I mentioned the most recent version of the pheno-age estimator, then the skin and blood estimator. I've worked with Michael Kobor's lab and Lisa McEwen on a new, what we call a pediatric buccal clock. So it's very accurate based on buccal epithelial cells. And others mentioned the clocks by Widener and Wagner. And so there are many clocks. And I just give you a comparison, because here I show you how these 8 different clocks perform in children. Why? Because that's where clocks break down. Many clocks work quite well in adults, but if you're interested in development, children matter. So these are children aged between 2 and 12, and you see my original clock in panel A, upper left panel, is very accurate, and so is my new clock, the skin and blood clock in panel D, the right. However, some of the other clocks that I mentioned are less accurate. Why? Because they were built in adults. Now here I compare these 8 different biomarkers in brain samples. So these are now brain samples from adults from 18 to actually 114. So they were brain samples collected from Steve Coles and the GRG, to their credit, and we analyzed them. And here again, the original multi-tissue clock in panel A works very well, very high correlation of 0.93. And the other clocks are less accurate in brain. When you look at the dots, you see some red dots, and it's not quite clear maybe on the screen. But these are cerebellar samples, And so it turns out that all epigenetic clocks underestimate the age of the cerebellum. And you can interpret that as a flaw of the clocks, or you can interpret this as a biologic finding. Because the cerebellar samples, in my worldview, the cerebellum is the part in the back of your brain. This part of the brain simply ages more slowly. That's my interpretation. And so I want to remind you, Steve Coles was the senior author on a study where we showed that, yes, the cerebellum ages slowly according to the epigenetic clock. And this figure was all created with centenarians and supercentenarians collected by Steve Coles and the members of the GRG. So this is the brain of a 114-year-old person. The upper panel shows you the ages, estimates from 30 different parts of the human body. And and again, on the leftmost, you see the cerebellum has the youngest age, you know. And
Do they
yes.
collect this after they die?
Yes, it's all post-mortem, yes. Yeah. Good question, yeah. So we can learn a lot from centenarian studies, I feel. For example, which organ age faster and not. And for these studies where you compare aging rates of different organs, you really need this multi-tissue age estimator, you know. Whereas when you have a blood-based age estimator, there are certain biases, and these analyses are conceptually more difficult to carry out. Here I show you a comparison of 3 of the widely used epigenetic markers, the multi-tissue predictor and also the predictor from Hannum from 2013. And I have all sorts of criteria. You see accuracy for estimating age in children, accuracy of estimating age in brain regions, correlation with gestational age, homogeneity of the estimates across different tissues from a supercentenarian, whether it relates to offspring status of a centenarian, cognitive functioning in the cortex, neuritic plaques, and so on. And on these metrics I just mentioned, the multi-tissue predictor, age estimator, really works very well and outperforms the other measures. However, when it comes to another outcome that we care about a lot, which is predicting time to death, we see that actually the Hannum predictor, colored in red, it's one of these panels, works well compared to the original multi-tissue predictor. However, the phenotypic age estimator, this novel biomarker we developed, it actually outperforms all other age estimators, and by a lot. It's substantially more accurate for predicting lifespan, as I mentioned. But let's talk a little bit about the biology. This radar plot also depicts how these different clocks relate to leukocyte telomere length. And you see that the phenotypic measure that works so well at predicting lifespan actually does have a stronger association with telomere length than, for example, Hannum, and the original multi-tissue estimator really doesn't have any relationship to telomere length. And a similar story can be said about naive T cells and other blood cell markers. So these some age estimators that were mainly built, were built based on blood methylation data, they tend to be confounded by changes in blood cell composition and leukocyte telomere length. That all makes sense, and it may even be desirable for certain applications, you know. Why? Because it measures immunosenescence, you know. However, the multi-tissue predictor is really separate from that part of biology. Moving on, I briefly mention our work on epigenetic aging clocks in mammals. So I want to develop epigenetic clocks for 50 different mammals, and this is a project funded by Paul G. Allen, one of the co-founders of Microsoft, and he has made a grant available so that we will profile tissues from 50 different mammals, every mammal we can get a hold of, you know, from horses, rabbits, pigs, cats, but anything. Why? So we believe that, of course, animals will be very valuable for preclinical studies. Many people focus on mice, but nothing wrong with studying other animals as well. And now, there are already epigenetic aging clocks for these different animals. So Matteo Pellegrini and Mike Thompson developed a clock for dogs. Another group developed by Jarman and Polanovsky, an epigenetic age estimator for humpback whales. And there are 3 or 4 papers that describe epigenetic age estimators for mice. And you heard earlier, Zymo also has a version of a mouse clock. Now, all of these publications, which are wonderful because they so much advanced our field, because they did show that gold standard anti-aging interventions really reverse epigenetic aging. So they, for example, calorie restriction or rapamycin or growth receptor knockouts, these interventions did affect the epigenetic age of these mice. Briefly mentioned, these are groups by Wolf Reek, Vadim Gladyshev, Trey Eidecker, Tina Wang, Peter Adams. They've shown that these gold standard interventions reverse aging, which is a great insight. However, there are challenges. And this is that all of these publications were based on a particular way of measuring methylation called reduced representation bisulfite sequencing. And it's the truth, and my lab also used it with Matteo Pellegrini. We have generated these data. And the truth is, it's been very difficult to compare these clocks across data sets. And why? Because these clocks often measure different locations in the genome. We heard earlier, a human has 28 million CPGs. So this technology may lead to high quality data in 100,000 loci. And then a month later, you repeat the experiment, you get another 100,000 loci, and there's no overlap. And so one solution to this problem is really to then develop a custom methylation array. In order to develop a clock that will work in many species, you want to focus on loci that are highly conserved across all 50 mammalian species. So I've had the pleasure with working with Professor Jason Ernst at UCLA and his doctoral student Adriana Sperley and Illumina, and we developed what we called a mammalian methylation array. And this array will profile 40,000 highly conserved sites in the genome. It will be applicable to all of these animals. If you happen to study white rhinoceroses, your research problems have been solved, okay? Or the microbat, so we can measure methylation in all of these animals, and the coverage is not great. This is you get 20,000 CPGs here and 20,000 there, However, it will be enough for measuring the ages in these animals. That it can do, It just isn't a genome-wide study. So are 40,000 conserved CPGs enough for estimating age accurately? The answer is yes. We've done a feasibility study with Michael Thompson has led that. So even using as few as 3,000 highly conserved CPGs, you can build very accurate age estimators in mice. Why study 50 different animals? We really need it for modern phylogenetic comparative approaches. We want to know why does a mouse live 3 years and why does a bat live 30 years, although they have similar sizes, So to do these phylogenetic comparative biology studies, you need large sample sizes. I want to briefly talk about telomere length. It's been mentioned several times that, yes, methylation age is much more correlated with chronologic age. The upper panel shows you how telomere length relates to chronologic age. And you see a big cloud, and the lower panel shows you on the very same individuals from the Framingham Heart Study do we observe much tighter correlations. But a more careful analysis really shows that epigenetic age acceleration, certainly of the multi-tissue clock, is not correlated with telomere length. And we know that from many very large cohort studies, thousands and thousands of people. However, Ake Lu, who sits in the audience over here, and she can tell you all about it. So she did a genetic analysis of epigenetic aging rates in blood. because just to tell you, one reason why some people age faster than others is genetics. There's no question. It's 40% of the variability in the ticking rate of the clock is genetic. So the question is, so what are the genes, you know? And so one of the loci that came out is very familiar to most of you. It's the telomerase locus, you know? And that is just mind-boggling, because if we had done a genome-wide study of telomere length, you would have found this locus. How do I know? Because the right panel shows the same graph for a genetic study of telomere length, you know. So you found telomerase. But the point is, we did the same with the multi-tissue epigenetic clock. We found the same locus, you know. Mind-boggling. Why is it? So this was utterly surprising to me. Why? Because it was a study of epigenetic aging, not telomere length. But there's another reason why it was so surprising, which is that the associations were so unexpected. Because imagine you inherit genetic variants that are associated with active telomerase. These very same variants that in certain ways are beneficial for telomere length, these very same variants actually are detrimental for the epigenetic age, meaning they are associated with faster epigenetic aging, okay? It was a paradoxical association. So just to caution everyone, we don't claim that longer telomeres are associated with older epigenetic age, because I told you earlier there is no relationship between telomere length and epigenetic age. But there's this what people call pleiotropic effect of this locus, the telomerase locus, the TERT locus to be much more precise. Telomerase is an enzyme with several components, so the TERT locus is associated, not the TERC. And here I want to show you some in vitro studies from in fibroblasts, again carried out by Ken Raj, where the red bars show you immortalized fibroblasts. They HTERT is expressed immortalized, and the y-axis shows you the epigenetic age. And what do you see? That with cell passaging, the epigenetic age continues to go up. So this is amazing, because even immortalized fibroblasts continue to age according to the epigenetic clock, again highlighting that these are different phenomena. Want to come to one study by Friedrich Stölzel and Martin Bornhäuser and the German team, who evaluated what happens if you carry out a hematopoietic stem cell transplantation. So unfortunately, some people who develop a certain type of leukemia, the AML leukemia, need to get their entire blood replaced. You know, so you then implant these stem cells. And the question is, imagine you're a 50-year-old and you get the stem cell transplant from a 20-year-old. And then 3 years later, what is the age of your blood? You know? And the study is very clear on that, without boring you, but it's all about the age of the donor. The 50-year-old with a transplant from a 20-year-old, his blood will be the age of the donor, meaning about 20-something, you know. However, of course, graft versus host disease then accelerates aging, you know. So this is, of course, a very dangerous transplantation procedure, has a lot of risks. But in principle, it's quite remarkable that even long term, if we followed this person, the 50-year-old who got the blood of a 20-year-old, and we followed him for 30 years, okay? And the question is, would ever the rest of the body reset the age of the blood? And the answer is never. It never happens. The blood will always have the age of the donor. You know, it tells you a lot about the biology of the clock. Many people talked about these lifestyle interventions. Everything that's known to be good for you, vegetable, fruits, exercise, it all has a beneficial effect according to the epigenetic clock, but I tell you the effects are very small. You need thousands of people to detect it. And so clearly what we want, of course, are small molecules and other therapies that have a much stronger effect on epigenetic age. And I'll stop with acknowledging the many people. I think I mentioned most of them. Thank you very much. Yeah. Yeah.
I've got some questions.
Yes.
One genetic change, or you mentioned the H-TERT locus kind of being associated with change, with differences in epigenetic aging.
Yes.
Somebody's asking about the biological significance of the genes that are found. And I know there's maybe a little bit of data about which genes are overexpressed or underexpressed due to those different CPG sites. Is there?
Yeah, that's a good question. More generally, what's the relationship between methylation and gene expression? And that's really a question that, in certain ways, plagues the entire field of epigenetics. We all have that question. There are some locations where you do see a relationship, but in most locations there is no relationship. And it's paradoxical. However, the people think that this effect could be not, the methylation might not relate to gene expression right next to the CPG, but perhaps further away. That's one line of thinking. And so yeah, I mean, I invite you, of course, to read the paper from R.K. Liu about this genetic study. It will come out in Nature Communications in a couple of months or a couple of weeks, I don't know. It's in the production phase. There's already an article on bioarchive, so you can, R.K. spent months, if not years, to carefully studying which genes are implicated, but just to warn you, it's difficult. Yeah. Yes.
There's a group, I think it's in Texas, that's looking at the telomere position effect over long distances, where they're looking at a loop of the chromosome on, loop of the telomere on chromosome 5, looping back to the H-TERT site. And they have fairly good data that nobody else yet has replicated, that telomere loop actually explains why telomerase gets reactivated when the telomere gets too short. And it probably has to do with CPG methylation and methyl binding proteins attaching to the sheltering proteins. Any comment on whether you think we may find more of these long distance effects of a methyl binding site?
Yeah, James Watson made a wonderful comment that telomeres loop back, and that might activate telomerase, and more generally long distance effects. I mean, I certainly hope that people carefully look at it, because this has stymied the whole field. How the methylation, or more generally even epigenetic changes, affect expression. Clearly, expression affects epigenetics. It's actually a two-way street. It clearly warrants detailed mechanistic studies. Yeah. Oh yeah. Okay. Yeah. I'll start. Yeah.
I really enjoyed the talk.
Thank
you very
you.
much. I think you made a tremendous contribution.
Thank
I
you.
would say that as somebody who focuses a lot on telomeres,
Yes.
I have to say that the HG Hutchinson-Gill report was very interesting. And when I look at telomere lengths, especially, I like to think of it in terms of, if you keep the telomerase active, then the telomeres not only get longer, but you have acquisition of other gene mutations, right?
That's right.
So the actual ability for stem cell-like cells to persist allows them to age in epigenetic terms, but their lineage doesn't die out. So part of the act of staying young is actually, as we see with the senolytic research, part of staying young is killing off these cells. So it's a double-edged sword. When you increase the survivability of stem cell lineages, then they accrue different
Exactly. So
heterogeneity.
exactly. I mean, clearly, people who fight aging, at some point, you need to maintain telomeres. However, what these studies show, that's not enough, because there are other things that continue to accrue, and people have done a lot of research. I mean, what we've shown here, the epigenetic aging continues to go on, unfortunately, you know. So I'm with you. It may serve a purpose. See, in my opinion, telomere shortening, you know, is clearly an anti-cancer mechanism, you know. But epigenetic aging could have a complementary role, you know. So even, in other words, if a cell is immortalized, then maybe these methylation changes serve the purpose to prevent further instability, you know. And
Well, I mean, as I understand the presentations that we've heard, the 3 amazing presentations, at no point are you saying that you're trying to understand the grammar or the actual meaning of gene expression. What I see is your data mining to find things that are highly correlative in a bidirectional way with measured age. But you're not making any interpretation as to what the grammar, the meaning of, or the stepwise expression of these lineages is. Because you're really just measuring differentiation of cells, and then you're assuming there's some hidden clock, whether it be how many cortisol squirts you've gotten, but you're not inferring any kind of mechanism here.
Well, I wouldn't put it this way. I would say the first step was to develop the most accurate molecular biomarker that's out there. That was step 1. But clearly that's not enough, right? We need to go much deeper to really understand, I call it the clockwork, the molecular clockwork. There are many thoughts. It could be circadian rhythm. It could be metabolic changes. You know, the genetic studies shed some light, but again, only the beginning. I think this is really the prize to understand the biology. You know, you've got to start somewhere. Yeah. Yes.
There was a question about DNA repair.
Yes.
And how that is linked with epigenetic aging.
Yeah. So DNA repair does have a relation to methylation, no question, in different ways. You know. My original paper from 2013 showed things like p53 mutations are associated with changes in epigenetic aging, you know. So there are certain indirect lines that sensors of DNA damage might be related, you know. But radiation treatment of cells, you know, which induces strong DNA damage, actually has not been found to be associated with increased methylation age, you know. So there's no easy connection. There's indirect evidence there might be, but who knows? Yeah, okay, James.
I know that a lot of your data acquisition or the training sets were based on the Women's Health Initiative. And, you know, the big controversy from the Women's Health Initiative was the effect of hormone replacement therapy in postmenopausal women.
Yes.
And that, you know, the data from that has been used as a club or a sledgehammer to alter practice patterns in medicine.
Yes.
So my question for you is, based on your data from the Women's Health Initiative, using either the multi-tissue or your new phenotype clock, do you believe that hormone replacement therapy, specifically in postmenopausal women, accelerates epigenetic aging? Does it have no effect, or does it decelerate epigenetic aging?
So the question is whether menopausal hormone replacement therapy has an effect on epigenetic aging, especially in the Women's Health Initiative. Now, the answer is complex. So first of all, in blood, we've never seen an effect. However, in other tissues we have buccal epithelium, you know. So we have shown that women who take hormone replacement, actually their buccal, the inside of their mouth, the buccal cells are younger, you know, than women who don't take that therapy. So I think probably a lot of anti-aging interventions will have an organ-specific effect, you know. Things might affect blood, but not liver, or vice versa, you know. Similar, obesity has a little bit of an aging effect in blood, no question, a little bit. But in liver, it has a huge effect, you know. But more generally about hormone treatment, I think this is clearly an exciting frontier. Like if that, I a lot of the data I look at implicate hormones in one way or another. And I wish somebody really investigated that in a very systematic way. Large number of people, different regimens of hormones, different sources of DNA, buccal cells. Why buccal cells? Because they have estrogen receptors, whereas blood cells have fewer estrogen receptors. So some organs are more susceptible. Yeah. Yes.
Have you considered doing the same kind of study on testosterone replacement therapy for men? I read that
I mean, yes, I've sent out lots of emails on that topic. The question is testosterone treatment in men. Absolutely, would love to study it. I mean, there are clinical trials going on right now, and my phone is always there. If somebody calls me up, I would love to analyze it. Absolutely, but not just testosterone, growth hormone, and so on. Yeah. Mm-hmm.
Alexei Alovnikov is the guy who proposed the existence of telomerase.
Yeah.
And his new theory is that although telomeres and telomeres are important for cell aging, he postulates that there's a hypothalamic clock and like a gravitometer that tells you how many moons you've lived, which is really out there. But I mean, there are organs that we don't know about, like the saccule,
Yes.
the organ we have for ultrasound. So
Yes.
if he's right, could you take some of the hypothalamic neurons and do a specific measurement as to the correlation between, like, a 20-year-old guy dies in a motorcycle accident, or a fetus is terminated at 18 weeks.
Yes.
Maybe looking at the hypothalamus might be where.
I agree. There's several
Even the
very.
hypothalamus has a lot of different
Yeah.
functions.
I mean, several people, very smart aging researchers, have voiced that idea. Study the relationship between the hypothalamus or function or various measures and aging in general, but epigenetic aging. Absolutely.
Across the 50 and see whether
Yes.
that's.
Yeah, I mean, probably the best way to do it is in animal models, right? So somehow to manipulate it, to see it in time courses, and really. So these are probably the most rigorous studies. And the good thing is there are already epigenetic clocks for mice and other animals, but I can promise you in a couple of months we will have super accurate measures of epigenetic age in animals, and then we can do it. Good idea. Okay. Yes. Greg? Yeah.
Speaking of, the hypothalamus,
Yeah.
there's a very exciting paper that came around August of last year from Kai's lab. He's been looking at the role of the hypothalamus in programming aging, and what he found is that a lot of that is mediated through neural stem cells in the brain. That their effects are mediated by exosomes.
Yes.
So I'm wondering, have you considered isolating exosomes from blood and running your clock on those to see if that would be a stronger indicator of what's going on in blood in general?
Yes. I mean, this was a very exciting paper, and absolutely. I mean, I think it could easily be exosomes, or James Watson always likes non-coding RNAs. It's totally on my radar screen, on many other people, because in certain situations, these non-coding RNAs have a strong relationship to methylation, especially at certain loci, Absolutely, it could very much be that. At some point, we looked at microRNAs. I remember in plasma, didn't find anything, but yeah. Who knows? Maybe one day somebody generates the data, and then there's a low-hanging fruit. That certain exosomes really coordinate the ticking of the clock. Very plausible. Problem is, many of us, we have already voiced lots of good theories. I mean, we just need data. Is it hormones? Is it circadian? Is it hypothalamus? So many things.
So more at the cellular level, somebody asked about, is there anything, has anybody upregulated demethylases and just watched what happened? And I guess maybe it's related to like the epigenetic age of a cell. There are things like reprogramming that obviously affect the epigenetic age.
Yes.
What happens when you tweak all the different pathways that Dr. Watson was talking about?
Yeah, so with my UCLA colleague William Yang, we will actually study all these components that were mentioned in mice. So these are conditional, these are knockouts, heterozygous knockouts. What happens if you knock out one of the DNMTs? And there are many in mice. What about if you knock out TET, So, and definitely we'll study that, There are already some people who have data on that, but I'm just saying that's what we will do in a few months, But right now I just don't know. Mhm. Yes, Greg? Yeah.
So,
Yeah.
the discordance between the positive effect of telomere length on the one hand and the negative epigenetic signature of that.
Yes.
Do you have any insight as to what causes that discordance? Are there specific sites that you can point to that are responsible
I think,
for that?
well, let me start by saying that it's actually not only at the TERT locus, okay? So these genetic studies have actually found many telomere maintenance genes, And so Arke has looked at several of them, whether she sees the same paradoxical associations. In other words, a genetic variant that is good for telomere maintenance, is that genetic variant then associated with increased methylation age? And although the findings are much less significant than at TERT, the same trend holds at multiple loci. So there is something about. It seems that things that are good at maintaining telomeres, whatever is good for that, is associated with older epigenetic aging. And now, why is that? One way to interpret these results, and Ken Raj has formulated that, is, well, if you have a cell that simply lives longer, why? Because telomerase is active, so it just lives longer. Well, then this cell has longer time for accumulating methylation changes. Do you see? And therefore, it makes sense that something that lives longer will have a longer epigenetic age. On that level, one could interpret that. That's one interpretation. Another is that at some point I postulated that the epigenetic clock relates to what I called the epigenomic maintenance system. That there's this maintenance system that plays a purposeful role. And another way to interpret these findings is that, well, whatever maintains telomeres is part of this more general machinery of maintaining the epigenome. But you had a thought. Yeah.
I've lost it.
Oh, okay. Wow. We lost other question. Well, thank you everyone for your patience.
Yeah, we owe special thanks to Bobby Brook. He's the one who made this happen tonight for all of us here in this room and at home, and those of you watching the recording later. So please, everybody, big round of applause for Bobby. And also for our speakers, Steve, Jim, Keith, all the time that it takes you not only to learn this stuff, but to put together outstanding presentations. Everybody, please. Now, we have learned the clock is ticking, people. So I hope all of this tonight is going to motivate you to take action to develop new therapies to turn back this epigenetic clock for lots more years of healthy living. So that's all. Thank you.